Good day, everyone. So today we'll be tackling about the structure and the process of DNA replication. So, what is DNA? Disoribonucleic acid, commonly known as DNA, is a nucleic acid that has three main components: a disoribose sugar, a phosphate, and a nitrogenous base. Since DNA contains the genetic material for an organism, it is important that it be copied when a cell divides into daughter cells. The process that copies DNA is called replication. Replication involves the production of identical helices of DNA from one double-stranded molecule of DNA. Enzymes are vital to DNA replication since they catalyze very important steps in the process. The overall DNA replication process is extremely important for both cell growth and reproduction in organisms. It is also vital in the cell repair process. So, why does DNA replicates? So, DNA is the genetic material that defines every cell. Before a cell duplicates and is divided into new daughter cells, either mitosis or meiosis, biomolecules and organelles must be copied to be distributed among the cells. DNA found within the nucleus must be replicated in order to ensure that each new cell receives the correct number of chromosomes. The process of DNA duplication is called DNA replication. Replication follows several steps that involves multiple proteins called replication enzymes and RNA. In eukaryotic cells such as animal cell and plant cells, DNA replication occurs in the S phase of interphase during the cell cycle. The process of DNA replication is vital for cell growth, repair, and reproduction in organisms. So let's go to the DNA structure. So DNA or disoribonucleic acid is a type of molecule known as nucleic acid. It consists of 5 carbon, disoribose sugar, a phosphate, and a nitrogenous base. Double-stranded DNA consists of 2 spiral nucleic acid chains that are twisted into double helix shape. This twisting allows DNA to be more compact. In order to fit within the nucleus, DNA is packed into tightly coiled structure called chromatin. Chromatin condenses to form chromosomes during cell division. Prior to DNA replication, the chromatin loosens, giving cell replication machinery access to the DNA strands. So, let's go for the DNA process. So, firstly, the preparation for replication, the step 1, replication fork formation. Before DNA can be replicated, the double-stranded molecule must be unzipped into 2 single strands. DNA has 4 bases called adenine, thymine, cytosine, and guanine that form pairs between the 2 strands. The adenine only pairs with thymine, and cytosine only binds with guanine. In order to unwind DNA, these interactions between base pairs must be broken. This is performed by an enzyme known as DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into a Y shape known as a replication fork. This area will be template for replication to begin. DNA is directional in both strands signified by 5 and 3 end. This notation signifies by side group in attach the DNA backbone. The 5 end has a phosphate group attached, while the 3 end has a hydroxyl OH group attached. This directionality is important for replication as it only progresses in the 5 to 3 direction. However, the replication fork is bidirectional. one strand is oriented in the 3 to 5 direction, or the leading strand, while the other is oriented 5 to 3, or the lagging strand. The two sides are therefore replicated with two different processes to accommodate the directional differences. So, next was the primer binding, where the replication begins. So, the primer binding. The leading strand is the simplest to replicate. Once the DNA strands have been separated, a short piece of RNA called a primer binds to the three end of the strand. The primer always binds as the starting point for replication. Primers are generated by the enzyme DNA primase. So, next was the step 3, the elongation. So, here was the process, or the primer binding. That first line. Okay. So, let's go to elongation. And enzymes known as DNA polymerases are responsible for creating a new strand by a process called elongation. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria such as E. coli, polymerase 3 is the main replication enzyme, while polymerase 1, 2, 4, 5 are responsible for error checking and repair. DNA polymerase 3 binds to the strand at the site of the primer and begins adding new base pairs complementary to the strand during replication. In eukaryotic cells, polymerases alpha, delta, and epsilon are the primary polymerases involved in DNA replication. Because replication proceeds in the 5 to 3 direction on the leading strand, the newly formed strand is continuous. The lagging strand begins replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerase then adds pieces of DNA called Ozaki fragments to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjointed. So step 4, the termination, So we have here an example. So the caption of the picture says is this polymerase blue attach themselves to DNA and elongate a new strand by adding nucleotide bases. So this blue heart-like thing was the DNA polymerase. Okay, so the last step was termination. Once both continuous and discontinuous strands are formed, an enzyme called hexanuclease removes all RNA primers from the original strand. These primers are then replaced with appropriate bases. Another hexanuclease proofreads the newly formed DNA to check and remove and replace any errors. Another enzyme called DNA ligase joins the Ozaki fragments together, forming a single unified strand, and the ends of linear DNA presents the problem of the add nucleotides and 5 to 3 direction. The ends of the parent strand consist of repeated DNA sequence called telomers. Telomers as the protective caps of end of chromosomes to prevent nearby chromosomes by fusing. A special type of DNA polymerase enzymes called telomerase catalyze the synthesis of telomere sequence that are ends of DNA. Once completed, the parent strand and its complementary DNA strands goes into familiar double helix shape. In the end, replication produces two DNA molecules, each with one strand from the parent molecule of one new strand. And that's all for today. So thank you for everything. I hope you learned something in this video. Thank you.